Hi everyone. Good afternoon. It's good morning for me, but okay. I'm really delighted to be able to share this work with this diverse audience. I'm sure you have had really wonderful time in the last week plus that you have had. And hopefully what I'm going to talk about will also be quite interesting. And if any of you get interested in working on this, please get in touch with me. My email is here as well as my blue sky account where I like the most. So my lab for mine has been interested in understanding odors or the smells around us. And there's a history to it, but we may have time to get there. So we want to understand why did the sense of smell or the olfaction evolve? Where did it evolve? And why is it evolving? So we use certain organisms to understand it. So a lot of biology. We look at humans a little bit. It's better to contextualize in terms of our personal experience. We look at mice as a model, and then most of the time we look at Sinorhabditis elegans, the worm that I'm sure you have been exposed to several times since your workshop began. And we look at how do they smell, odors which come from bacteria around them often or from plants. So odors are made by two big branches in life. They are either microbes or they are plant in origin. So flowers and so on. For the purpose of this talk, we'll focus mostly on microbes and then see where does neuroscience come into picture. So you are familiar with lots of smells that you see on the slide. So I'm on slide 2 if you're in case you can't see it. So some of the smells that we're very familiar with, and you have probably smelled them since you were a child, is a smell which is called dimethyl sulfide, dimethyl disulfide, dimethyl trisulfide, dimethyl pentasulfide, and so on. So all of them contain sulfur attached to carbon units, for example. So this is in cabbage. This is in lots of other things, for example, in eggs and so on. And we are very sensitive to responding to this. We don't know how our nose smells it. We don't know the receptor or sensor for this, but I'll tell you a little bit about how we might solve that problem. The, and this is a smell that you might dislike, but something that you experience quite a bit. On the left, you have this odor which may be very familiar to all of you. So if you have ever been to a movie theater and you have bought popcorn, what you smell is called 2,3-butane-dione. We will use the term diacetyl for it. And this is a double ketone that is used as flavoring agent. It's also present in curd, in beer, and everything else. And at very small concentration, it's very pleasant. And again, we can smell it really well, but we don't know how we smell it, on which part of our nose allows us to smell. And the third one, which is again a very common molecule, is amine containing molecules, and this is called trimethylamine or TMA. And this can be called a rotten fish odor, and not just rotten fish make it, but lots of other organisms when they're dying do it because there are bacteria which are metabolizing it and produce lots of trimethylamine. Other amines, for example, phenylmethylamine or phenylethylamine, are also present in the urine of lots of predatory animals. For example, all the big cats have phenylethylamine in their urine, and this is how they mark their territory. This allows other predators to know that here is a boundary that I shouldn't cross. It also lets their prey know that there is a predator around. So there's a large family of receptors called TAAR or triethylamine receptors, which are present in the nose, especially of small rodents, which allows them to know here is a predator and I should stay away. Um, so these are just examples of a few that you have smelled since you were born, but we this idea that how we understand it and go to network level analysis of connectomes is just catching up. One of the smells that I really like, and I really like this smell is called geosmin. or petrichor. So this is the smell that we all experience if you are in India when the monsoon rain falls. So you might, if you are from North India, it hasn't happened yet this year. But this is the smell called geosmin, which is produced by some of the fungi whose spores are lying dormant in soil. And when the first rain falls, it releases this odor. And this was quite some time ago. And recently, the idea that petrichor exists and its use in research has taken up. Again, we don't know how we smell geosmin, but we know how we smell this in a fruit fly called Drosophila melanogaster. So we know that we have very strong responses to odors. We perceive them as bad odors, for example, smell from your armpits if you haven't taken a shower for a while, from a garbage can, or good one, for example, in a flower, or maybe a perfume that you use, or just from a baby. Um, and all these are happening because we have hundreds of or about 400 receptors, which are represented in the nasal epithelium, which allows us to smell them. That goes to olfactory bulb, which is um, part of our brain from the nose to the olfactory bulb, and then it goes to olfactory cortex, which is here. And this processing obviously has layered meaning, but we won't get into that. Now, when we lose this ability to smell, which can happen either due to defect in our nose, in the olfactory bulb, or olfactory cortex, we have conditions called anosmia or hyposmia. Uh, and you have become familiar to these terms, or many of you would remember these terms, because about 5 years ago, when we, at the start of COVID-19 pandemic, this was the first symptom which appeared in many people. They lost their sense of smell either completely, so it was anosmia, or they lost their sense of smell partially, and it was hyposmia, and they gained it at a rate which was really very individual. So we need to understand why, how do we gain this sense of smell? How do these circuits function? And how can we gain it? So although this is not a predominant feature now in even people who get SARS-CoV-2, there are conditions which are congenital anosmia. Anosmia as well as hyposmia is about 95% prevalent in people who suffer from neurodegenerative disease, such as Parkinson's disease, and some cases of Alzheimer's and of amyotrophic lateral sclerosis, but most commonly in people who have Parkinson's disease. And many of you would know people in your family who would have Parkinson's disease. So, how do we begin to understand how we smell it, what are the sensors in our nose which allow us to smell, and how do we understand the circuit? So I'll begin at the proximal question as to how do we understand, how do we smell certain smells. And so based on a large amount of work which was done in the last 40 plus years, we know that a membrane receptor called G protein coupled receptor um, allows us to smell smells or odors. And these GPCRs can have very different kinds of smells. Ligands. They could be odors, so volatile molecules, but they could also be soluble molecules. So a subset of G protein coupled receptors for whom the ligand only is a volatile molecule is called an odorant receptor. So all odorant receptors are GPCRs. They are expressed in our nose, but in other places in our body, and we get there. And they allow us to respond to certain odors. So for example, this specific molecule, if it was present in one of the cells or a number of cells in our nose, was bound to a smell, for example, diacetyl or the butter popcorn odor, it will engage other molecules downstream of it and then cause changes in the cell, which allows it to talk to the next neuron and so on till it reaches olfactory cortex. And this is very well understood in many cases, and we'll see examples of how this technique can be used to understand this. Um, so a lot of it is understood in humans, for example, at least at the level of what we can smell, but we don't know what their receptors are. So humans have about 400 odorant receptors. Majority of them, about 20 of them, express really highly in the nasal cavity, but we don't know what their ligands are. So about about 8 years ago, there was a woman called Joy Milne who came up and said she can smell people who have Parkinson's. So this is Joy now. This is Joy when she was married about 40 years ago. And she lives in a place called Perth, which is about 30 minutes from here. And she could smell people with Parkinson's because her husband, who was a doctor, developed Parkinson's, and she could smell everybody. So she went and knocked at the door of a doctor in University of Edinburgh and say, "I think I have a special nose and I can smell Parkinson's." So they started a study, got funding, and they allowed her to smell clothing from people who had Parkinson's. So there was a group of people or cohort of people who have had Parkinson's, but she was also allowed to smell people from clothing from people who didn't have Parkinson's or had no evidence that they had Parkinson's. So they had no tremors, no gait difficulty, and no other things to suggest that they had Parkinson's. And she faithfully picked 45 out of 45 people, so two-thirds of 45 out of 45 people who had Parkinson's. She also picked up a 46 person who was from control group. Turns out that person developed Parkinson's the following year. So she was able to smell Parkinson's before it developed, before the gait symptoms or neurological symptoms developed. So this has initiated studies, several studies in the UK, where she's a participant. So she sits alongside a machine called gas chromatography mass spectrometer, or GCMS, the kind that my lab has as well. And then she does better than the GCMS. Really fun person to talk to and very inspiring. This is a digression, but just to say that because of one person like Joy, there have been several research projects which have been funded, and it has spearheaded lots of research in this area, supported also because of arrival of COVID-19 and hyposmia and so on. So she is what is called a super osmic person. Um, Before we move on, I also wanted to describe another called phantosmia, when you think you smell something when the smell is not present. And this phenomena is not really well understood. So while I was talking about there being receptors in our nose, which allows us to smell lots of beautiful smells around us, and we can do it for billions of smells. There are receptors called odorant receptors, or OR, which are present not in our nose, but in lots of other interesting places. And these are very interesting. So, for example, we have them in our liver. So these three receptors are expressed not in the nose, but they are expressed in the liver. This specific one is expressed in the kidney. These four in the pancreas. These in the adipose tissue, or the fat that we collect around our middle of our body, and these in our gastrointestinal tract, or the small intestine. Why is this subset important? These are the tissues which allows us to metabolize or store glucose, and these are the organs which lead to lots of lifestyle diseases. For example, type 2 diabetes, obesity, fatty liver disease, and so on and so forth. All the things which are increasing in number all over the world, but specifically in India. So why do we have a smell receptor not in our nose but in our gut? Why do we have them? And this is a question that I'm personally very interested in. We don't have data yet, but we are making some progress towards that. The other little thing that you will see is a sperm. This is human sperm, and this human sperm expresses 20 different odor receptors in different parts of this tiny little cell, which is a cell body, has its nucleus, which is going to fertilize, and its flagella. why does it have 20 different odorant receptors? And this is something that uh, we as a lab are very interested in. Not only because it's special to human, because if you start looking at sperms from other organisms, for example, rats and mice, they have the same 20 receptors, and we don't know what they smell. But something that if Sita had this this meeting again 2 years later, maybe we'll have something to say. But really exciting that smell is um, the ability to smell is retained not in our nose, which we use on a daily basis, but also in other organs. Um, so coming back to our nose and part of our brain which processes odors, there are lots of very interesting studies that I wanted to bring up just to make it really exciting. And if any of you are thinking about looking at it later in future, you may find that these are very interesting ones. For example, there's a lab in Israel who looks at how odors present in tear or human tears affect behavior. And they use both human and mouse models to study this. For example, this study is one of my favorites, is that chemical signal, especially a volatile organic compound in human females, can lower aggression in people around them. They did really cool, innovative study to look at this. Most of the other ones are looking at how does it affect health, how does it affect obesity. For example, in mice, you can give them fatty food and they gain weight. So for example, the one on the right. And both of these are given the same high fat diet, but the one on the left didn't become fat only because its olfactory bulb was removed. So your ability to smell obviously changes your ability to metabolize and store lots of carbons in fat or glucose or in carbohydrates and allows you to become fat. So this is a very interesting idea that if you can smell better, you can eat better. And that might be able to tell you why some people who cannot smell do not eat poorly and they become they suffer from a condition called anorexia, which is quite common amongst teens, but also in people who have neurodegenerative disease. If they can't smell, they do not eat well, and they do this. Lots of other ideas, lots of other tidbits of information that people who have variation in one of the receptors in our nose called OR74, there is a cohort of people in Canada where they have variation in this receptor, and again, they have different obesity profile than others. So this has become a target to understand, can we smell differently? Can we change our ability to smell and eat less? Many of you are probably aware of a drug called Ozempic and two other variants, which makes you feel full and so that you don't eat, and it works differently. But the idea is that if you can have a combinatorial treatment so that you don't eat well, eat more than you need. Coming back to odorant receptors, which are present in our nose, which are sensory neurons. This is one of my favorite slides, which doesn't feature my favorite organism, C. elegans, but it looks at all other placental elements as well as some other vertebrates. So, for example, you see humans, they have about 414 odorant receptors. They have just three ethylamine receptors that I talked about, which are amines secreted in the urine and other organisms. What is really interesting is that while humans have 414, if you looked at something much bigger, for example, a cow, this number grows really high to about 1200. In mice, we have about 1100. In rat, about more than 1200. And if you were to go look at something which has a much larger nose, you will think of an elephant. It has about 2000 odorant receptors. Why does it need 2000 odorant receptors? We don't know. The idea would be that it requires them because it has retained them, simply because we know that if we don't need something, these genes undergo variation and they're not selected for. So we observe this phenomena called pseudogenization. That means the ones on the left are the true genes. That means they are made into proteins. They are expressed in some of the cells in our nose or in other organs, while the blue ones are called pseudogenized, that means they're not expressed well. So lots of our genes have become pseudogenized, that means they're not expressed while some are retained. And this subset, which is expressed, is of uh, interest because if we can figure out what they do, what they allow us to smell, we can begin to understand this. So as a lab, we are interested in understanding the function of odorant receptors and also to identify ligands for odorant receptors. And I've been talking about what the model could be, and I have given it away already. But you could study them in octopi. They are really beautiful model because they have on these suckers lots of odorant receptors. Although they're in water, they are actually odorant receptors. And they do really well at smelling. Not a really good model in the lab. And then the elephant, which is about 2000, it's really nice to do that, but hard to study. And then you have mouse. And then we come to C. elegans, which is, for reasons that you know already, probably in the last few days, has become a really interesting model for us to study. It has about 1,400 GPCRs. That means they're not just for odorants, but for other molecules. Um, so we decided that this is what we'll go with. And while we are trying to figure out what each one of those receptors might do, we have been looking at what their ligands could be. And this is a really understudied area of research simply because people who try to identify odors or the ligands for olfactory or odorant receptors are all in food industry. So they look at when they make orange juice, do they want to spike it with a flavor called limonene because that's what reminds us of lemon or citrus flavor. Or if they make beer, they want to have enough of diacetyl so that it's at a perfect level where we say, okay, this is beer, this is what I like, this is where I want to spend my money on, and so on. So one of the things that we want to do is to start very systematically look at who makes odors, what is the most abundant odor, which ones are likely to be most relevant to humans, to mice, to C. elegans, to Drosophila, and across the wider world. But we need to pick some examples. So what we know based on what is available in literature is that volatiles or the molecules which can become volatile are the ones which are produced by the cells. They are often very diffusible so that they can go out of the cell. So they are in water or fluid phase, which is aqueous in nature, but they are very short and they have um, chemical groups such as acids or carboxylic acids, alkenes, alkanes, aldehydes, ketones, alcohols, or so that they can really very quickly partition from an aqueous phase to a vapor phase. So they can keep going as soon as you make it, you can smell them, right? So we know that for them to go partition from aqueous phase to a vapor phase, they need to be small, so typically less than 300 Daltons, and they often have one of these uh, functional groups, right? So we said, okay, we are going to start looking at who makes them and go after each one, right? So back to C. elegans, we we have looked at C. elegans, which is a lab strain called N2, and again, you have been probably been exposed to it several times by Abhishek, by Sitabra, and other people. So for this purpose, we took a strain of C. elegans, which was isolated from a mushroom not far from here from a place called Bristol in UK. But we also looked at a number of strains which are come from other continents, and I'll have a little bit to say about that. So the reason we think C. elegans relies on a sense of smell quite a bit is that they are found in rotten fruit. For example, this orchard from our collaborator in Paris. And once they've consumed part of this apple, they really need to get to this apple. And they can't get here if they can't smell which way the apple is. This was the start of a really interesting collaboration some years ago. And for me, thinking about why olfaction should be important. So again, if this is a repeat, bear with me. C. elegans hermaphrodite, which means it has both eggs and sperms, has about 959 cells in its soma. So the big neurons and lots of little neurons that we're interested in, and 302 of these 959 cells are neurons, and which are labeled with a green fluorescence protein in the worm at the bottom. Now, we are interested in a very small subset in the head ganglia here, and they compose what is called an amphib sensory organ, or their nose, so to speak, nose and mouth. Because of lots of work done by lots of different people, but initiated by Sidney Brenner and John White and completed in 1986, they were able to create what is called the mind of a worm. And this, you would have seen variants of this image several times. And here is the neurons that we are interested in, so infant sensory neurons. And I will not talk about connectometry, but we can discuss it at the end of the talk if you are interested. So how do we begin to look at it? So a couple of years ago, or about 3-4 years ago, um, just when we're trying to get out of the pandemic and do some of the work, um, the C. elegans, all the C. elegans individual neurons, all 302 of them were sequenced individually, so one at a time. And this data was made available to the entire community. And you can go and explore this. So for each one of these neurons, so these are the olfactory neurons that C. elegans has. So it has only 6 olfactory neurons. So in its nose, it only has 6 cells. And they are called AWA. There are 2 of them left and right, AWB left and right, and 2 AWC neuron. They're called on and off. And we won't go into why they're called on and off. But using this data, Ritika, who was just joining the lab then and she's ready to graduate now, was able to look at this data and say, these are the receptors which are expressed either in this, or this neuron. And there were some which are expressed in all of them. But it was very interesting to see that there were some G protein coupled receptors expressed only in this neuron. And this is the neuron which allows worms to smell diacetyl, for example. This one for smells that worms hate, and so on. So it says, okay, we are going to go after these 474 receptors and then see if we can find out what they smell. It seemed like a tall task, but we have begun to do this and see. So one of the first things that we wanted to ask is to connect our study to something that worms have to experience every day. So if you remember the picture of C. elegans in a rotting apple in a orchard, or it could be a rotting banana tree, for example, if you were in India, because we have been able to find them on the campus of IISc when we were looking for them. So 2 questions that worms have to deal. Worms are very interesting because they are hermaphrodite. They don't need to find necessarily a partner. So we can turn that part of equation off. And then say, what do worms care about? Which kinds of smell worms care about? So you need to do 2 things if you're a worm. You need to find food. So you need to find bacteria that worms eat bacteria. They need to find good bacteria, which are good source of calorie, good source of proteins, carbohydrates, and fats, and micronutrients, for example, vitamin B12, and other vitamins as well. Or they need to avoid pathogens which might produce toxin and kill them. And these are the same pathogens that affect our health as well. So for pathogens for C. elegans are Pseudomonas aeruginosa, those who cause lung infection in us, for example, in cystic fibrosis or pneumonia. Or these could be gastrointestinal pathogens, for example, Salmonella enterica, which causes dysentery, and so on. So because of work from lots of different labs, including ours, we have already set a large established that C. elegans is killed by toxins of human pathogens. And one of those pathogens is Pseudomonas aeruginosa. It's a gram-negative bacterium. It's part of escape pathogens. So B is for Pseudomonas, which is rapidly evolved antibiotic resistance and has a broad host range. So, for example, it infects C. elegans, but it can also infect obviously our lungs. But it can also cause urinary tract infection. It's a major pathogen in diabetic foot ulcer, burn wounds, any skin wounds that you have. So this is a major pathogen, and it is a pathogen for C. elegans. So we decided to see if worms can smell Pseudomonas aeruginosa. So I will turn off pointer and then run this movie, which was done by Deep and okay. And the movie doesn't work. 1 second. I'm going to escape and see if okay. I'm trying to see if the movie will work. If so, what you would have seen in this movie is that worms were placed on a lawn of Pseudomonas aeruginosa, and they would have walked away. And this we could measure and then see why. And so the question that we asked them was, why did they walk away? And we looked at the olfactory and gustatory neurons of C. elegans. So again, taking you back to what is on in the amphid neurons of C. elegans. So this is 12 pairs of neurons, of which 3 pairs are olfactory neurons, which are shown here. Turn on pointer. Um, so these 3 are olfactory neurons, AWA, AWB, and AWC, that I referred to earlier. And Deep was able to ablate all of them by a method called cell killing. And then he found that only when these 3 were not allowed to function, worms did not respond to Pseudomonas. So they couldn't walk away from Pseudomonas if these neurons, which are shown here, were ablated, killed by a method called laser ablation or genetic ablation. So another movie, but I'm not sure it will work. Oh, this works. Awesome. So what you see on the left was is when birds were on the lawn of Pseudomonas on the left, and we placed them in the center of the lawn, but they walked away. What we're looking at them is they're out of this, away from this bacteria as much as possible. So that was good. So this is what we were able to measure in this. How much time do they spend with the bacteria and how much time do they spend off the bacteria? And on the right is a mutant of C. elegans, which cannot smell. So this is an olfaction deficient mutant called ODR3. And what you will be able to appreciate is that those worms remain on the lawn. They can't walk away. And they can't walk away because they can't smell. So this was very exciting for us, and this was several years ago when we were in Bangalore, and this was the first student who says he's going to take this observation and then follow it through to figure out what is the neuron, what is the receptor, why do we smell it, and can we identify what this odor is. So Deep was really, he did beautiful experiments where he says worms which cannot smell Pseudomonas also died sooner, so there is a positive benefit to your ability to smell. So this is when they decided that, okay, really cool movies that he could make. And then he says, okay, this is the time where our lab has to move into understanding odors or volatile organic compounds. And then we decided to do what is called gas chromatography. And this is a method which allows you to take very small amount of odors from very small number of very little samples, for example, a few million bacteria, and then say what is it that they're making. And this takes advantage of a process where bacteria or any other organism, for example, we do it from rotting fruit, for example, you allow them to build what is called a headspace. That means you put them in a container and all the volatiles will go from aqueous to vapor phase, collect and make what is called a headspace. Headspace is also something you would have heard if you drink beer. And Then you take this headspace and use an absorbent material, for example, in this really little needle, which is available from companies like Agilent or Waters, collect those volatile material, and then you send it to a GCMS, which has multiple components. But basically, what you're doing is that you're heating this adsorbent material again to do what is called thermal desorption, so that you release what you had adsorbed first, and then you fragment these ions. So you take the mass of what you had collected, then you fragment it further, and then look at mass by charge ratio to identify what each ion could have. And then you compare what is coming out of the GC to look at what it could potentially be by comparing it to a library of chemicals which is available to test against. So by lots of trials and errors, because we were new to the field, we figured out how to do this. And when we were able to do this, we figured out that everything smells. For example, plastic smells plastic. Any plastic container that you use ever has lots of smell. For example, polystyrene. So you'll see lots of styrene. You have stabilizers that you can see, and this is what you have in the top slide. So all these peaks that you see are examples of polymeric substances and their degradation product, which are contained in plastic material of any kind. What you see at the bottom is what Pseudomonas aeruginosa was making, and Deep was able to identify 6 different molecules, which was really nice. So DMS and DMDS is the sulfur-containing molecule. This is in cabbage as well, but then we saw lots of other ones. We were able to set up assays called chemotaxis, and again, you may have been exposed to this, where you ask worms to choose between a test chemical and a solvent where it was diluted, and see what do worms do. And I'll show you some movies in a bit to see how they respond. But within about 15 to 30 minutes, worms will say, oh, I like this test molecule or I don't. And if they like it, they will go left. If they dislike it, they will go right. And we can calculate something called chemotaxis index. So if they don't like it, it will be a negative chemotaxis of as high as -1. We were able to say that of these 6 molecules that Pseudomonas made, one of them called 1-andesine, which is an alkene, was a repellent. And again, show you a movie. This is where worms were doing chemotaxis. What you see in the center is worms. Here is 1-andesine, and they would studiously avoid this part of the plate, but they will mill around over here. If you look at ODR3 mutant, which is on the right, they cannot differentiate between the site containing 1-andesine or the site containing control, so they spend their time all over the plate. So this again suggested that olfaction or odorant capacity of worms were important for us. They did lots of other experiments. Again, worms are quite human in the sense that if they don't like something, they turn away, and which is what you'll see on the left. They placed a drop of 1-andesine, and one says, oh, no, I don't like it, I'm going away. And they do something called an omega turn, which is really curious motion, involves lots of different body wall muscles to execute this. So they did it so that they can go back instead of going forward. The worm on the right is ODR3, and same experiment, except ODR3 didn't realize that there is an offensive chemical, and they pass through the liquid. So again, to reiterate that worms really need to smell to be able to get there. The really set up lots of different assays to look at this behavior, which is also very common in neuroscience. You look at both behavior and neuronal signaling, and he looked at how do worms move in an arena where on the left there was no 1-andesine, but on the right there was 1-andesine. So what you will appreciate is that worms on the right, where 1-andesine was present, worms spent a lot more time roaming around. They were not able to settle in one place and eat, which will look like this. So worms were not able to settle down and eat because an offensive chemical such as 1-andesine was present. Um, so how do worms smell this? And this, to look at this, we created ablation for all the olfactory neurons one at a time. So ODR3 that I mentioned lacks all three neurons, the functionality of all three neurons. Now, Deep created ablation of individual neurons, AWC, AWB, AWA, and then found that only AWB neuron, which is shown here in red, was not able to smell it. So which was nice. To confirm that this was indeed true, Deep had to establish what is called an essay that you would have come across probably in talks from Abhishek or in other neuroscience talks, probably from other organisms. This is where you look at signaling downstream of a G protein coupled receptor in the neuron. So this is what you see here is is the soma of AWB olfactory neuron. And this is expressing a molecule called GCAM. So this is GFP, or the green fluorescence protein, fused to calcium binding portion of another protein called calmodulin. So that protein is responsive to calcium. And when there is increase in calcium, this GCAM will fluoresce better. So left versus right. So the right one is fluorescing more. So when a GPCR is bound by a ligand, and this stimulates activity of trimeric G protein, which is composed of three units called G alpha, G beta, and G gamma. And if G alpha is the stimulatory kind, calcium level will increase, and you might have understood this already, but if not, please ask me at the end of the talk. And as a result, if the calcium increases, there is increase in GFP of the GCAM, and you will see that the signal will increase. So this is for each worm. When worms were exposed to 1-andesine, GCAM was present in AWB. So initially there was no increase in calcium, but when it was removed, you will see that each one of the neurons is firing and there is increase in calcium, right? So they suggested that AWB neuron was the neuron which was able to respond to 1-andesine signal. So this is good. So we started with an odor which was not known to be smelled by C. elegans, and we were able to trace it to one of the neurons called AWB. We were able to also go a little bit forward, gratifyingly, and say that 1-andesine is a signal which allows worms to perceive this as a pathogen and allows them to enhance their immune response. So sort of like how a vaccine would work. So this is called priming of immune response. So if the worms were allowed to smell 1-andesine early on, and they can now survive better when Pseudomonas is presented. We do a lot of biochemistry and molecular biology, so we were able to identify a mutant of Pseudomonas called PA145320. This is how the genes are named. Nothing to remember. Now we call it UNDA locus because it's easier to see. And if you compare the top with bottom, both of them are Pseudomonas aeruginosa, except the one on the top lacks UNDA gene, and that's why you don't have this 14-minute peak, which is 1-andesine. So we went further on with this and then say, okay, is 1-andesine acts like a vaccine or primes an immune response? And Deep did really beautiful work of allowing worms to smell 1-andesine for 2 hours, and then say, what does it do when you smell something that you don't like? I think as a human, if you smell something that you don't like, you probably can walk away, and which is exactly what C. elegans was doing. But is there a physiological response? Are you doing something else which will prepare you for an for a response to a pathogenic bacteria, which is what was happening. So if not only you can change your behavior, but if you can also upregulate an immune response which will protect you in case you are infected, for example, that's a beautiful thing, and that's what you want to have in something which acts like a vaccine. So to do this, Deep allowed worms to sniff one anisine for 2 hours and then say what is happening transcriptionally. So he looked at genes which are upregulated when to protect worms from Pseudomonas aeruginosa, and he was able to show that 3 different genes were upregulated, some of them really highly, and then it protected the worms from subsequent infection with Pseudomonas. And what was really gratifying is that when worms were allowed to sniff one anisine, they were protected from Pseudomonas aeruginosa, but they didn't induce signals from, but not from other pathogens. So it was a specific response. Again, owed to what we can do with C. elegans. We used a reporter. Again, this is IRG1, one of the genes which are upregulated by the odor, and it gets turned on when worms come in contact with Pseudomonas. So no GFP when Pseudomonas is present, but GFP also when only the odor is present. So this is the first example where we are saying we know there's a pathogen when we can smell a specific molecule from the pathogen. So this is akin to saying that if you can smell limonene, you can say, oh, it's citrus flavor. Maybe there's an orange or oranges close by or grapefruit. Or you can smell, you can take phenylethylamine and then one of the rose odors and then say, oh, there are roses close by. Um, so I'll bypass some of these and then move to why is it important. Um, After moving here, we have been doing some of the ideas. Can we use some of these orders to develop diagnostics? So these are clinical strains of Pseudomonas aeruginosa, which also make this odor called B, which is monoamnicine. So this presents a really beautiful opportunity to see if we can create a sensor, maybe an electronic nose, either using metal oxide framework or some of the cool nanodot technology to see if we can detect Pseudomonas if somebody has Pseudomonas in their lungs. This is work in progress, but this is just to show that we have been able to get to a place where we can detect it in clinical strains, which came from lots of different individuals with varying clinical conditions. So to come to the second part, and I'm taking too much time, so I'll rush through this. The second part was to see, do worms only recognize offensive odors? Do they also use olfaction to find something that they like? So we are looking at whether they use olfaction to find food that they like. So another movie, if I can play it. So here are worms where they're given 2 choices. One on the right called OP50 or E. coli on the left is a bacterium that is found in its gut. So it's part of what is called microbiome. So for those who may not be familiar with microbiome is a community of bacteria which lives in different parts of our body and utilizes nutrition therein. So for example, there is those in our gut which harbor there. So this is called gut microbiome. You have a different microbiome in our armpit. We have a different one in our oral cavity or in your mouth, and so on. And for example, you would have heard that microbiome helps us digest certain food that we don't have enzymes for, and that's true. But in the last 20 years, this field has told us that it's very important for our health in many different kinds. So we have been looking at microbiome of C. elegans, and this is one of the bacterium called CEP1 from the gut microbiome of worms. And as you can see, this is just tracking using a MATLAB code that Ritika has written to show that worms within 5 minutes make a decision that they like this new bacterium more than the E. coli food that we usually provide, not tasty at all. How do worms recognize this? And this is the work done by, started by Ritika and shown by Nikita and other people, Angus here. And so they looked at 12 molecules, 12 bacteria which are in the gut of C. elegans. And they resemble the gut microbiome of humans, not exactly by name, but what they can do. And then we asked worms which ones do they like and which one do they not like. So of these 12 bacteria, worms said, oh, we like these 3 bacteria. And so the next question we simply asked was, do they like these 3 bacteria because they smell different? Do they smell nice? And if that's the case, what is the smell that they like? So again, back to our technique of gas chromatography or GCMS, we looked at all 12 bacteria and found that, the 3 bacteria that worms like indeed produced a lot of a molecule called isoamyl alcohol or IAA. Then there are other molecules as well, but it was easy to see that isoamyl alcohol made up most of that. Ritika and Nikita showed that both of them, all the bacteria produced more of isoamyl alcohol if an essential amino acid called leucine is present. So leucine is an amino acid which is the most abundant amino acid in our proteins. And we know that proteins are what you see most of us. So proteins are the building, not only they're the functional units, but also add to the mass of our cells. And they are the functional units. They're enzymes, they're receptors, and so on. So it says, okay, we need leucine. So maybe worms want more leucine, so they want to have those which produce isoamyl alcohol, because isoamyl alcohol is coming from leucine. So Ritika was able to figure out that this is the pathway which worms use to make isoamyl alcohol from leucine, and she was able to perturb this pathway to say, okay, yes, this is the pathway that these bacteria use to make isoamyl alcohol. Since we're running out of time, I'll go quickly through this. Ritika and Nikita used a set of mutants that we had generated for AWA, AWB, and AWC neurons, and looked at all the molecules that these microbiota made. So we looked at each one of them individually and then say, do worms like them? If they like them, there'll be positive chemotaxis index here. For example, if you see it here, that means worms like this when leucine is present, and so on. They also use the same chemotaxis assay to say worms like butyl acetate, phenylethyl alcohol, IAA, methyl isovalerate, and so on and so forth. And using an ablation of this olfactory neuron called AWC, they were able to say that almost all the molecules produced by microbiome are sensed by AWC olfactory neurons, and just one of them by AWA olfactory neurons. So this was interesting because so far most of the attention for odor research has gone to AWA neurons. So at this point, we also wanted to see which one of these smells are important for worms. So we went and looked at a large number of isolates or natural isolates of C. elegans, which are from different places, which are marked in colors here. I guess the farthest away was one in New Zealand, and there's one from Chile, and lots of them from Europe. All we ask these worms is to say, do they like isoamyl alcohol? How much do they like? So if you see the numbers at 1, they really like it. So every worm says, I'm going to go, I want isoamyl alcohol. As a control, we used acetoin, which was one of the molecules sensed by another neuron. And then they say, oh, I like it, but not so much. I can live without it. So this data was very important for us to say that isoamyl alcohol is an odor which is in the nature very important for worms to be able to find, and that's why they haven't lost their ability to sense it. And this became important if I go 2 slides later. So it says, okay, we have identified the neuron. Can we identify the receptor or G protein coupled receptors? So this is where Ritika went back to this list that she has created and then say, I'm going to look at these 29 receptors which are here in AWC neuron, or these 15 which are in other AWC neurons. So we collaborated with a lab in Sweden, so Changchun Chen at University of Umeå, who at this time was creating GPCR edited lines using CRISPR, so 6 at a time. And we decided to work together to see what they are. So we got all the strains from Changchun Chen, so that we have about 298 edited strains who cannot do olfaction, but we didn't know what is it that they don't. And this is one piece of data where if you look at this strain called 1169, it doesn't have 6 different GPCRs, but it could not respond to isomal alcohol well. So then we mutated each one of the 6 GPCRs one at a time, and then Ritika found that one of them, called SRD12, shown in red here, could not recognize ethanol alcohol. Um, and this is just an example of how worms respond to isomal alcohol. So this is timer in minutes. Within 3 minutes, wild-type worms said, oh, I love isomal alcohol. I'm going there. Every one of them went there. However, so this is a trace for how wild-type worms behave when isomal alcohol is presented. And this is a trace for one of the SRD12 mutants. And as you can imagine, they're going everywhere but where IEA is. So they think there's no smell present. So this was our first foray into looking at this deletion library that Changchun created, and we were able to identify this. So, I'll bypass this, but the idea here is that C. elegans, based on these 2 studies that we have done and others in progress, looks like a really interesting model to identify receptors for odors which are very common in the environment of C. elegans. And using genetic tools both in C. elegans and in bacteria, we're able to see how this odor, for example, in this case, isomal alcohol is generated. How does C. elegans perceive it using an olfactory neuron and a specific receptor, but also be able to provide why should it matter to worms. Now, are these important in humans and mice and so on? And to get to this, this will be the second last slide for this so that we can discuss a bit, is that we have been looking at what kind of receptors are there in our gut and the gut of mice or rats, and there are many, many of them. And most of these are names of the receptors. And they remain names because we don't know what ligands they have. Some of these are olfactory receptors. So to get to this, we really need to create what is it that the gut can potentially smell. And this potential smell in the gut has to come from gut microbiome. And to do this, we have one way to look at this is to look at what kind of smells that the feces or our poop contains. And we've been doing this with mouse feces. And this is just an example of feces from mice. You will see names called butanoic, hexanoic, bentanoic acid, and esters and indole, which is made by, for example, E. coli. But what you see at 4 minutes is isomal alcohol. So the isomal alcohol that we discovered based on work from C. elegans and its microbiome is also relevant to mice, and now we know human by looking at a subset of them. So we have taken one of these receptors from mouse gut, expressed in C. elegans, so that now it's expressed in AWC neuron. And when we do that, worms can better smell its ligand called butyric acid. So this is again ode to C. elegans to be able to say that when we have a really interesting model system, it not only it gives you a chassis to understand how proteins in humans or mice or anybody else that you're interested in might function. And so I'll stop here and summarize to say that odors or volatile organic compounds are produced by almost all microbes. Some of them are very specific. For example, one andesine is only produced by Pseudomonas aeruginosa. Worms are really great model to understand olfaction biology and neuroscience behind it. although I didn't go into that a whole lot. They perceive some odors as repellent and some as attractant, and this is similar in all organisms, including humans. Some repellents are perceived as pathogen-associated molecular patterns, and these are really important to identify because we can use them to induce a favorable immune response. While the attractive odors are signals for nutrient-rich diet for worms. For example, for if you are somebody like me, I have been doing GCMS to look at very complex food and then see what is the smell that I like about, let's say, just fried samosas, for example. I won't tell you what that is, but if I meet you in person, I might. Um, what this leaves us also is to figure out. I have only told you the first part. I have stopped at olfactory sensory neuron, but because we know the connectome in C. elegans, we know who the next potential partner. In these cases, they are interneurons, which are either wired through synaptic connection or gap junctions. What they are. And at some point of time, if we had interested people who want to come in and trace that, we would be very interested in understanding what they are. We'll stop here. I've taken too much of my time. My current lab members and a few more, and the previous lab members at IISc. I want to thank University of Dundee, the Royal Society, and Philo Scotland for current funding, and welcome to DBT India Alliance, which supported bulk of funding where we started this lab work in Bangalore. Thank you so much. I'm open to questions.
Questions? I understand there were a few questions during the talk which weren't audible probably. So, Varsha, can you hear the question?
Yeah, I can hear you. Go ahead. You bet.
So, is there any form of cooperativity between these receptors? As in, do these receptors interact?
Interesting idea. We do not have any evidence that they interact. The idea perhaps is that they do, again, not work from C. elegans at all. We have not looked at them or the strains that they have. We are looking at one receptor at a time. But we know that odorant receptors occur in rafts, and there is some evidence that odorant receptors and ion channels co-occur. So there is some cooperativity there. But having two different receptors working together, we have no evidence in C. elegans. But I would not be surprised if that's the case. We have been doing a little bit of work on how selective the odorant receptors are for their ligands. So part of this is looking at alpha folds to see how big their ligand binding pocket might be, how much variation that they can accept in the ligand. For example, if you have a 4-carbon ligand can be extended to a 5-carbon backbone, and will the same receptor still work? The idea is short answer right now based on our work with 2 different receptors is yes, but if you go beyond 6 carbons, then they will stop responding, and then we envisage that there's another receptor, whether that's in the vicinity or elsewhere, we have absolutely no idea. And as far as I know, there is no lab which is looking at it per se. But thank you for the question.
I just had one more question. The AW neurons that you mentioned, the 3 neurons, are they specific to different volatile chemicals, or do they also do some form of combinatorial?
Right. Okay. This is, we do not have the answer, but I try to attempt it. So, bacteria. So obviously the single odor at a time is a lab concept, right? Worms are in the nature are assailed by lots of different odors. Even from a simple bacteria that likes, for example, scentworm. The movie that I showed, it makes 5 different odors. It makes 7 different odors, but 5 of them are attractive odors. It just so happens that isomalt alcohol is the most, it takes up the most space. It's about 40 to 60% of the head space, but the other ones are important as well. So we have done some experiment to see if all of them are present, do worms get there sooner? it seems like that's the case, but we have not been able to tease out the contribution of each one of them. The combinatorial becomes more important if you have, for example, a bacterium which is making odor, which is sensed by AWA, but also by AWC neuron, and how does it play out? We have done a different variation of the experiment where we say, okay, here you have a bacterium which makes an attractive odorant, for example, isocyanated alcohol, but also makes another bacterium which makes this attractive odorant as well as a repellent molecule, and we can do that by microbial genetics. Worms within minutes very quickly figure out the difference between these two bacteria, and very little difference in gradient. Obviously, both AWA and AWB neuron in this case are firing, but they are both wired to 3 different interneurons. So the idea to test that will be to figure out which one of these interneurons are working and how do they talk to command interneurons, which makes the decision whether to go or to run away. Again, these are ideas that are very clear-cut, should be done, but haven't been done by our lab or any other labs, but there are labs in the US who are in the best position to answer some of these. Hopefully they will do them.
Any other questions? So, Varsha, I mean, I'm curious, and mainly this is connected to Harish's question. Like, I asked you this, like several years back. Like, unlike Linda Buck's work on the olfactory system, where we know there's a combinatorial port, in the case of C. elegans, is it clear what kind of strategy they are using to recognize all these various types of odors that they should be able to recognize?
No, the short answer is we do not know. For example, so again, I'll give an answer which is not the most satisfying. We have taken 2 bacteria, both of which produce isocyanated alcohol, but they also produce a different subset of other attractive odors. And in that case, one seemed to trace differences between isocyanated alcohol, which suggests that certain odors are predominant, and worms pay more attention to, for example, isocyanated alcohol than to other attractants. Ideally, this should be followed up with GCAMP study to see in what sequence they are firing, when they are happening, but we do not have answers to that. I think, Sitanga, I wanted to actually pose another very interesting question that C. elegans poses, which is very interesting, is that we have 600, we have only 6 neurons, we have 474 odd receptors. How does C. elegans generate this diversity of responses to, at least we know right now about 135 odors that individually worms respond to? How do they do this from 6? So the question from, we have too few neurons, but we have to generate a variety of responses. How do worms do that? And that's a very interesting question that worms can be utilized to look at.
Yeah. I mean, which makes you believe that there must be some kind of a combinatorial code, isn't it? Like,
yes, there must be. There must be. So one of the ideas is that the second layer of GPCR signaling is to start looking at G alphas. So worms have about 22 G alphas, two betas and gams. So that's not where the diversity is generated. But each one of their sensitive neurons, so if you just look at F8 sensitive neuron, each one of them have about 5 or 6 G alphas. And we don't understand the combination of GPCRs or individual GPCRs with each one of the 6, for example. Do they always couple with the same G alphas? Do they couple with different ones? And how is this coupling affecting response that's generated afterwards? It's easier to understand locomotion, for example, by GCAMP, but then there are long-term responses, for example, in physiological response or transcriptional response in rest of the soma that we don't look at all. So it will be interesting to see how that generates diversity. There are about 30 odd regulator of G protein signaling called RGS proteins. Nobody looks at them. And I'm sure they're part of the equation of how you generate diversity and then combine it, right? There are 2 parts of the problem.
Yeah.
So very interesting ways that one can look at, and I think firms has to look at them. I think so there should be funding to look at them and to do them.
Right. Okay, so let's thank Varsha for this fantastic talk, and